All right, everybody, this is MPM 2020 again, and this is Boris Riepen from Philadelphia, and we are really excited about tonight's keynote session, and with so many fantastic talks and posters today about organelle biology, about the apicoplast, the mitochondrion. So this really was a great lead up into our keynote presentation. And I will turn it now over to David Roos from the University of Pennsylvania to introduce the keynote speaker.
Thank you, Boris and Nina and Manoj for the invitation to introduce today's keynote speaker, Akhil Vaidya from Drexel University College of Medicine. I think Akhil requires no introduction to most longtime participants in the molecular parasitology conference. Akhil is, I believe, I know for certain Akhil was at the first MPM meeting because I was there with him. And I believe that Akhil and Diane Worth are probably the only people who have attended every single MPM meeting. That's not quite true. He wasn't there in person for the meeting that was supposed to begin on September 12th, just after the World Trade Center attacks. But he had abstracts in that book. And in fact, if my count is correct, tallying the Vaidya Lab's contributions over the past 30 years of MPM, this year marks the year of your 100th lab abstract in the Woods Hole meeting. So congratulations to Akhil and the Vaidya Lab. It's a little bit daunting to think about introducing Akhil for some of the obvious reasons. Certainly, you want to make sure that you don't get things wrong for such an important member of our community and distinguished scientist. Also for other reasons. Many people know that I have been fairly outspoken in my criticism of the keynote speakers series. That I have felt that the presentations and often the introductions don't live up to the quality that we've come to expect from the posters and student and postdoc presentations at the meeting. But I will do my best not to disappoint from my part, and I'm sure that Akhil will follow on from the really spectacular talks that we've had thus far. You can see the title of Akhil's presentation listed in front of you, Stumbling Towards Truth, Basic Research Guiding Anti-Malarial Drug Discovery. And as I thought about the title and things that members of his lab, present and previous, were kind enough to share with me, I realized that you can view this title very much in the context of what I might think of as the Vaidya dialectic of confusion or constancy, the confusion implied by stumbling and the constancy applied by truth. This confusion starts even as early as his name. I trust that Akhil's parents and family call him Akhil, but from our community, he's more commonly called Akhil, and Akhil, to his own credit, has even been known to introduce himself as Akhil. So I believe it's more accurate that the pronunciation Akhil is shown over on the left. What is clear, that's not confusing at all, is that the term Vaidya means a doctor, and Akhil comes from many generations of physicians in his family. He was born in the state of Gujarat in the region of Saurashtra. Here's a picture of Saurashtra, well known as the hometown of Mahatma Gandhi. And Saurashtra itself is an area with a lot of confusion and a lot of constancy. Having holidayed there myself, I can attest that it's a place of inspirational learning and history in the temples of Somnath and Dwarka. Despite being in a dry state of Gujarat, it's also the home to resorts and casinos in the former Portuguese colonies of Daman and Dieu. Looking online from a picture of that, this picture of the spring break student chugging something was viewed as an advertisement for coming there, probably not what the state of Gujarat wants to highlight. And it's also the home to the factories of Reliance Industries, which are the closest thing I've ever seen on earth to something that looked out of a Mad Max movie. So an area of both confusion and spectacular interest. Akhil was born in 1947, also a time of confusion in India, leading to the state of India today. 1947 was the year of Indian independence. He was one of three boys in the Vaidya family. I will leave it to you to try to discern which of the brothers shown over at left is Akhil. And he has, despite many years of scientific work in the US, remained close to his Indian connections. Those of you who have traveled in India will recognize that a common scene on the highways of India can be represented here. I should say this is not Akhil on his way to school on the left. And I'd encourage you to look up the YouTube video showing the family at the right with not one, but two dogs on their motorcycle with the entire family. Akhil has tried to replicate his heritage in his lab here in Pennsylvania, as you can see in this picture with his lab. I don't think he could fit them all onto his bicycle. Further manifesting the confusion or constancy of the Vaidya scientific trajectory, he moved to Bombay for his studies at Bhavan College at University of Bombay, where he got his PhD as well in 1972. Of course, Bombay is now Mumbai, and he then moved to the US to work at the South Jersey Medical Research Fund, which has now become the Coriell Institute, and then to a faculty position at Hahnemann University, which became the Medical College of Pennsylvania Hahnemann, which became Allegheny University, which became Drexel University College of Medicine. And Akhil can correct me if I'm wrong. I don't think I've missed anything, but there may be something else involved in that history as well. Akhil jokes accurately that he's one of the few of us who has maintained his entire academic career in the same office, but he has been in 4 or 5 different institutions despite never changing his office. Now, we're here not just for the history of the speaker, but for the science that he embodies. And I took the liberty of pulling out a couple of my favorite aspects of Akhil's scientific career, those that have been most influential to me and I think the field as a whole. And when Akhil shared his presentation the draft presentation with me, I found that he was going to talk about most of these. So I'll just highlight a couple of points. Many people in the parasitology community do not realize that Akhil got his start working in virology, working in particular on murine memory tumor virus and seeking to try to identify human tumor viruses. It was a logical hypothesis that the important role of MMTV in breast cancer in mice might message the important role of a tumor virus in breast cancer in humans. Some of his first publications involved the search for a human breast cancer virus, publications in Nature. These themselves were quite important papers at the time, but probably key to his critical and insightful understanding was the recognition that, as we now know to be the case, tumor viruses are not responsible for breast cancer in humans, and recognizing that initial hypotheses might be wrong has been a hallmark of Akhil's scientific career. That was particularly true in the work for which he's best known in the parasitology field, studies on the parasite mitochondrion. The Vaidya lab was responsible for identifying repeated elements in Plasmodium DNA, which then were revealed to be part of an extra chromosomal DNA, a linear 6 kb molecule that encodes mitochondrial functions of Plasmodium. The Vaidya lab was the first to recognize this, despite several labs, at least 4 labs, independently identifying and mischaracterizing what we now know to be the apical genome as the mitochondrial genome of Plasmodium. It's not. The mitochondrial genome is a tandemly repeated 6 kb linear molecule that was first identified by Akhil and students and postdocs in his laboratory. Many studies over the years have gone on to elucidate the molecular and cellular biology of the mitochondrion in Plasmodium, including recognition of the inheritance as for all endosymbiotic organelles, showing that both the mitochondrion and the apical genome exhibit uniparental inheritance during sexual replication in the mosquito, identifying key aspects of antimalarial action that target the mitochondrion, including the synergy, the molecular basis of the clinically important synergies between atovaquone and proguanil, and in more recent years, elucidation of a number of important drug targets and mechanisms of action focused on the mitochondria, but not limited to the mitochondria. Shown here are some of the papers that I believe Akhil will mention in the course of his talk. So I won't dive into the science now, except to cite that in keeping with his retention of his historical roots, I was interested to note from Akhil's CV, as I had known from discussing with him, that he's retained his ancestral familial heritage in Ayurvedic medicine, publishing papers with his brother, characterizing, for example, the anti-parasitic activity of traditional medicines in India. So a couple of stories that are worth highlighting that indicates both Akhil's connection to his Indian heritage and also his dedication to science. Not surprisingly, many of the stories that members of his lab were kind enough to share relate to the central role that the Woods Hole meeting, the molecular parasitology meeting normally held in Woods Hole, Massachusetts, plays in our life. Several people from Akhil's lab reported that on the long drive from Philadelphia to Woods Hole, he thought it would be useful to recite some of the many things that he has memorized over the course of his career. But there is some confusion among people in the lab as to what these recitations are. Some people reported that Akhil was citing 1,000 generations of history of his family in medicine, which seems a little hard to believe. I think more likely is the recitation of the 1,000 incarnations of Vishnu, which I'm sure must make for a entertaining road trip in the 5.5 hours it takes to drive from Philadelphia to Woods Hole. Other stories relate to the to Akhil's engagement in the party scene at Woods Hole, and subsequently one former colleague recalls sharing a room with Akhil at Woods Hole and being awoken in the middle of the night to an intense and passionate scientific discourse on endosymbiotic organelles and inheritance in Plasmodium, only to recognize that this lecture was being delivered by Akhil in his sleep. All of these point to the central action of the central importance in Akhil's work, of both work on Plasmodium and of members of his laboratory. This is a word cloud of publications from Akhil's lab, and you'll note the prominent role of Plasmodium falciparum, despite Akhil's longstanding jealousy for those of us who are fortunate enough to work on better biological systems like toxoplasma, the parasites shown in back and on my t-shirt. But more importantly, the members of his laboratory have been instrumental for all of the work that they've carried out. Mike Mather, Joanne Morrissey, many others shown on the slide. We have numerous pictures of laboratory parties including those at Woods Hole, shown down below, as well as laboratory activities that involve axe throwing contests in Philadelphia. Such is the role that he plays with members of his laboratory that he's clearly been involved in other important events as well. And this applies not just to his laboratory, but as I can attest as a frequent visitor to the campus, and it's a role that from the outset, Akhil has been a core member of the microbiology and immunology department at Hahnemann or Drexel or MCP or Allegheny or whatever it is. This picture I love is so very 1970s. I love the Polaroid photographs, the pinking shears, the plaid shirts, and most of all, the bad hair. Clearly, Akhil, you could have done something better than this. But he has certainly been instrumental in fostering the students and postdocs, but also members of his department. I will point out that, for example, in a department of 35 faculty, 6 of them work on malaria. Here is long-term collaborators, Burns and Bill Bergman. And just as a final illustration of Akhil's commitment to the field, several years ago, in the early days of the Plasmodium Genome Project, Matt Berriman and I coordinated an annotation jamboree in Hinxton, UK. You can see Akhil here, hard at work on the Plasmodium genome. And only when he was there did I learn in an email from his wife that we were there on Akhil's 60th birthday, which is how I know exactly when it was that he was born. And you'll notice Akhil with many friends of the MPM conference. I think I can recognize Manuel Linas and David Fiddick and Bob Sinden and Brendan Crabb and Malcolm Gardner, Jennifer Wortman, Chris Newbold, Dan Neefsey, all of whom I'm sure are either present as participants in the MPM meeting that we have been holding this week, or perhaps maybe watching this event as a live streamed YouTube video. So with that, let me turn the screen over to Akhil and tell you we're looking forward to hearing your stumble towards truth, basic research guiding anti-malarial drug discovery. Thanks very much.
Wow, I can't believe this, how you put this all together, David. Thank you for the introduction. Yeah, you've been a critic of these keynote addresses for a long time, and you did a great job of introduction. I'm not too sure how I'll do with my talk, but we'll figure that out. So we'll see how that goes. And thank you very much, Boris and Manoj and Nina, for your invitation to come and give this talk. It's an honor, and I greatly appreciate this. This is the 31st year of MPM, and I look forward to many more to come. It's going to be great fun.
Sorry, Akhil, just to avoid distracting you too much, I will shift from the picture of endosymbiotic organelles, mitochondria included in toxoplasma, to a picture of Plasmodium, just to...
It's about time. It's about time you do that. Thank you. So let me share my screen. As this title indicates, so it's really great to be participating in this from my kitchen, essentially. But as always, it's really great to see so many early career parasitologists attending this virtual MPM. So I thought it would be appropriate to recount my own meandering journey, even though David tried to give some introduction to that, but it has been a meandering journey in science, and how I stumbled through 50 plus years of being in academia. I started my graduate school in 1967. I was 19 years of age at that time. So it's been a long journey. And to tell you some of the a few kernels of truth that were sort of revealed to me So it may appear that this is really a long time to most people, but for me, this has been like a flash. I can say honestly that now, though, is really the most exciting period of my career. I'm really looking forward to what it might reveal in the future. So I really I expect to come and attend MPM for years to come. So I see no real retirement in future for me. So Albert St. Georgi, a preeminent biochemist and Nobel laureate of the last century, said when he was he was asked to write a prefatory chapter for annual review of biochemistry, and I'll paraphrase him here. He said, the cheapest form of one's own funeral is when one carries a candle and walks to the graveyard. Given this keynote address feels a bit like that, but I hope the funeral is some distance away. So thank you. So getting this started, this is the sort of outline that I will be talking about from virology to parasitology, milkman to malaria. I'll talk about what I call Polaris and other things. So as I said, I started graduate school when I was 19, and my advisor really gave me a lot of freedom, and she was not a virologist, but I wanted to work on viruses that cause cancer, and she gave me freedom to work on that. And one of the things that we were trying to look at, and she had a collaboration going on with Dan Moore, who was over here in US, to look for mouse memory tumor virus related particles in human milk. So my job was to actually, one of my jobs was to go around looking for human milk. So I started as sort of a milkman, going around asking, convincing women, nursing women, to donate some of their milk so that we can analyze them for virus particles. So that work actually led to an interesting paper. My first paper was in Nature, which, I mean, I didn't expect that to be the case. I didn't realize what Nature was at that time even. And that was the first author is, of course, and by the way, this was published in 1971. It was Dan Moore, he was on my thesis committee, but he also became my postdoc advisor when I came to US. And I was the last name. I had probably minimal things to contribute to this paper, but they were kind enough to put my name on that. And as David mentioned, that the whole idea about human breast cancer virus related to mouse memory tumor virus is actually not borne out to be true. So I always say that the only really good thing that came out of this paper was my then girlfriend and my now wife was impressed by this paper that I published and how the publicity that it got. So Bill Weidance was a professor in Department of Microbiology at Hahnemann when I came to Hahnemann as a faculty member, a junior faculty member. And Bill was a very well-known immunoparasitologist, and Bill worked on malaria. And he said, oh, you should do something about making genomic libraries from which we can fish out antigenic genes, the genes encoding antigens, and we can make vaccines and all sorts. Oh yeah, piece of cake, we can do that. So he started doing those things, and he convinced me to start working on malaria, and I've always been thankful to him for that. He passed away in 2017. We miss him very much. But another person who was also part of the department was Carol Long. And Bill convinced her as well to work on malaria, and we actually formed a malaria group. And so malaria was something that we started playing around with it in the early 80s. And as David mentioned, that I have my ancestral origins are from Vaidyas or the doctors, and he's my great-grandfather, Madam Vaidya. He was one that who actually sold anti-malarial Ayurvedic formulations, and that was one of the reasons why I felt that I was sort of continuing that. And this was laid down to me by my brother Akhil Vaidya. Here he is with his turban. He doesn't usually wear a turban, but I have a picture of him in turban. And he told me that, what are you doing working with this mouse memory tumor virus? You know, what does that have to do with humans? You should be working on something important. Work on malaria. This I started with. And then that actually also was an induction for me to start doing more seriously working on malaria. And so the first paper this was one of the first papers that we published on this tandemly arrayed gene clusters of malaria parasites. So in the library that genomic library that we had made, we just wanted to make sure that the library was of good quality. So we probed it with total genome and fished out what appeared to be repeated sequences. And those repeated sequences were then subcloned, and I sort of started working on that from time to time. And it turned out that these were highly conserved across different species. And Shabadi, Berghii, Falciparum, Nosy, Sanomolgi, all of them had this sequence, and they all added up to about a 6 kb in size or so. And this was highly transcribed as well into different RNA molecules. So this is about the time when the people were working on different potential vaccine candidates, antigen genes, and the Australians had a really great knack of coming up with acronyms for these antigens, such as FIRA and MISA and RISA and all that. All these are really great names. So I thought, we are no fools. We should name this as something important as well. And I named that as Polaris. Polaris means Plasmodium origin large array of relatively invariant sequences. I actually tried to get this name in publication by writing in the manuscript that we submitted to Molecular and Biochemical Parasitology. The editor, I think it was George Cross, was the editor at that time, and George said, "Oh, this is too frivolous. Take it out." So it unfortunately never really made it to anything. So Polaris, as we sequenced it and all that, turned out to be the mitochondrial DNA. Mitochondria, and that has essentially occupied my intellectual life for a significant amount of time since then. I like to point out the Sanskrit word. Those of you who can read Devanagari Libri, you can see what it says. Matru Kendriya. Matru means mother. Kendriya means center. Mitochondria being inherited by through maternal lineage. This seems like an appropriate thing to say. So we sequenced this 6 kb DNA. It turned out to have mitochondrial proteins and portion of ribosomal RNA, and so on. And just about that time, as David said, there were several people who were working on what they thought was the mitochondrial DNA of malaria parasites. And here is a paper from Malcolm Gardner when he was working in Ian Wilson and Donald Williamson's lab. They published about the same time, calling this the mitochondrial DNA, and that actually turned out to be, as you know, the apicoplast DNA. Okay. Here's something that we sort of stated back then, saying that all this information, the amount of all the information that we had from 35 kb DNA molecules, tempts us to speculate that the 6 kb molecule is a highly derivatized mitochondrial DNA, that 35 kb molecule is a highly derivatized chloroplast DNA, and that these molecules are expressed in separate compartments. We have no evidence of what those compartments were, but we had some idea what they may be. By extension, this speculation could also be applied to other organisms of phylum Epicomplexa. It will be important to investigate these suggestions further, because if true, they may affect our view of evolutionary position and modes of control of these important pathogens. I sort of feel proud of putting this sentence in there. We had no idea what the speculation was it's poured out to be true. This is in 1990. So this is something that has continued. Well, clearly, I've decided to leave the epicoplast to all the epicoplast people who've been, a lot of that we have not done much work with epicoplast at all. We focused on mitochondrial DNA. So mitochondrial DNA is really bizarre, really weird. It codes for only 3 proteins in Plasmodium, as well as most other epicomplexins. And it has these ribosomal RNA genes in pieces. So at that time, I was, we had figured out that there was an RNA world before it became the DNA and protein world, and that RNA has enzymatic activity and all that. And when I saw all these pieces of ribosomal RNA in both strands and in a scrambled fashion, I said, that's a possibility that these actually could be primordial ribosomal RNA catalytic sites. Could that be the one that would be? So we really spent a couple of years trying to see if there are any catalytic activities associated with this ribosomal RNA. Turns out it was totally wrong, and I really apologize to Mike McIntosh, who worked on that in the first 2 years of his graduate school on that. It turned out not to be the case, but we know that there are many these ribosomal RNA gene pieces that are present on that, and Gene Fagan actually has mapped them out very carefully on the mitochondrial DNA of malaria parasites, and how they actually come together is something that really is an amazing thing. So Swati Das just talked about this in earlier session today. These RNAs don't come together and form a single piece. They are not joined together. Instead, they remain as single small pieces, and they are somehow coming together to form these kind of structures that can be formed. And how they are coming together is still a big problem, and that's something that we hope Hang Jun and her collaborators will eventually figure it out. Other people may want to work on that as well. So given all this stuff that I was really transitioning from virology to Plasmodium, and this whole ribozyme-associated activity of these RNA pieces were not going anywhere, I decided I needed to go on a sabbatical. And I did that in 1993 and went to Tom Wellham's lab. Along with David Caslow, I worked between the two labs. It was a great sabbatical. I really made lots of friends over there, and also it was scientifically really rewarding to me. We showed during those times that mitochondrial and epicoblast genomes are maternally inherited, as David mentioned, and that there is a unidirectional mating that, like I said, only in certain strains there is, when certain crosses, only one parent acts as a maternal parent and the other one does not act as a maternal parent. We have some evidence that there might be some this hierarchical mating patterns, and which may be indicative of early stages of speciation. That's something that's really worth looking into, and I think would be interesting to follow through. So what are the functions that are critical for mitochondria? We know that generation of electrochemical potential is the key to mitochondrial functions, and that drives a lot of the mitochondrial physiology. So TCA cycle will then generate these reducing equivalents that allow the electrochemical potential to exist, and that happens through the electron transport chain pumping protons across the inner membrane. And the proton gradients is then used for synthesizing ATP, and there are other functions that are associated with mitochondria in general, synthesis of various metabolites such as heme, steroids, iron sulfur clusters, nucleotides, and so on. So we wanted to see what are the functions that are critical for malarial mitochondria and so on. So after a few years, one of the things that we actually figured out was that the TCA cycle is totally dispensable in blood stages of Plasmodium falciparum. But it is critical for insect stages. So in blood stages, TCA cycles, 6 out of the 8 TCA cycle enzymes can be knocked out in blood stages. And overall, the central carbon metabolism in the blood stage Plasmodium falciparum is highly flexible. Okay. This work actually was done by Hang Jun Kai and Ian Lewis, Ian, when he was in Manuel's lab. There's a collaboration that really was really great to work with. So it was really fun when they were at Princeton at that time. So this has been published a few years back. It's sort of interesting analysis of genetic knockdowns and its impact on different metabolic fluxes. So other important function that people thought would be very important was the heme biosynthesis. The heme biosynthesis pathway in plasmodium falciparum is divided between epicoplast and mitochondria. Some of the reactions occur in mitochondria. The initial reactions, the first reaction and the last reaction occur in mitochondria. Others occur actually in the epicoplast, and at least one in the cytoplasm. Hangjun was able to knock out ALA synthase, and parasites were fine. She knocked out ferrochelatase, and parasites were fine. And this in the blood stages, and she knocked them out both together, and parasites were fine as well. And this actually was shown in this paper to show in collaboration with Dan and Carol Long and with Paul Segala working in Dan's lab at that time, that the biosynthesis was essential only for the development in mosquito stages, but not in the blood stages. So there's something that actually turned out to be a really great finding, because the heme biosynthesis for a long time was considered to be a potential target for especially since it is uniquely distributed in different compartments, that it was considered to be a potential target for antimalarial drugs. But this work showed that it's most likely not that good a target. Okay. So mitochondrial electron transport is the other part that's critical for as a canonical function of mitochondria. And you can see that you have in plasmodium falciparum, the electron transport chain is somewhat abbreviated. There is no complex 1, but there is a single subunit NADH dehydrogenase, and there is dihydroorotate dehydrogenase and 3 other dehydrogenases that all create reduced quinone. These quinones will have to go through oxidation through BC1 complex, the QCR or complex 3, and regenerate oxidized quinone to be serving all these different dehydrogenases. And electron transport chain continues, and so on. So what could be the functions for these things? Well, it turns out that the electron transport chain was likely to be inhibited by certain antimalarial drugs. And the sequence analysis, when we gazed at the sequence carefully, we saw that cytochrome B had some really interesting features. So I'm not going to bore you with the details of the Q cycle, but there are 2 different centers in the BC1 complex called QO center and QI center. QO center is where the ubiquinol gets partially oxidized 1 electron at a time, and QI center is there's another oxidized quinone is present. It gets reduced over that. So this sort of is the Q cycle is critical to that QO site and QI site, and you can see that QO site sequence in plasmodium falciparum was relatively well conserved, but the QI site was significantly different. And based on these sequences, we predicted that these would be the reasons why certain antimalarial drugs such as atovaquone will be working in a selective manner, essentially working as a cyanide for malaria parasite, inhibiting electron transport in malaria parasite without affecting our own mitochondria. And it's something that's been interesting to look at. Well, we wanted to then do some biochemistry with mitochondria and malaria parasites. Those of you who have gazed at malaria, blood stage malaria parasites, it's hard to actually find out where the mitochondrion is. Here's the mitochondrion. If I did not label it with M and having a 2 double membrane over here, you would not even consider it as mitochondria. It's a really wimpy mitochondria, and but it is still essential. It's minimal but still essential organelle. And we wanted to figure out how best to really study this. And Indra Srivastava was in the lab at that time, developed this really very nice assay using flow cytometry to measure membrane potential, and he also carried out respiratory measurements of mitochondria as well. And he showed that atovaquone, which is a reasonably broad spectrum antiparasitic drug, works against toxo as well, collapsed mitochondrial membrane potential, and that happened in a dose-dependent fashion. All the other drugs don't do that, and it also inhibited respiration as well. So having these this assay actually allowed us to do further investigations as well. But what's the interesting aspect about the electron transport chain was, what is the main function? Why you need that? If ATP synthesis is not a major source, if glycolysis is the major source for ATP in malaria parasites, and this was something that was published a few years back, work by Heather Painter and Joanne Morris. Joanne is the sort of pillar of my lab, and along with Mike Mather. What we tried to look in this was that we knew that pyrimidine biosynthesis is a critical part of mitochondrial function. So if you have dihyroorotate dehydrogenase, which is present in mitochondria, that has to be the ubiquinol, which is the electron acceptor for this enzyme, needs to be oxidized in order for this reaction to continue. So if you look at dihyroorotate dehydrogenase, DHOD, you see that it catalyzes the fourth and the only redox step in pyrimidine biosynthesis. Okay, and most of us, humans and plants and gram-negative bacteria, they have this what they call type 2 DHOD, which is membrane associated, and it utilizes quinone as the electron acceptor over here. And that reduced quinone over here needs to be reoxidized in order for this reaction to continue, and the continuous production of orotate, then making pyrimidines coming from that. Okay, but bacteria, some of the bacteria as well as yeast actually have what are known as type 1 DHOD, and that takes fumarate as the electron acceptor in the same reaction, converting it to succinate, and you can generate fumarate coming from various other sources, so you don't need to really recycle succinate to fumarate, but you can have fumarate coming from other sources that will provide continuous feeding of the metabolites to go to pyrimidine biosynthesis. So what Heather and others, what they did was to actually make a transgenic parasite in which yeast DHOD, which is a type 1 DHOD, fumarate acceptor was introduced into Plasmodium falciparum. These transgenic parasites were expressing this gene, and not only that, these parasites became resistant to atovaquone. So here is the wild-type parasite. Atovaquone works against the parasite growth at this level. While if you added just one metabolic bypass, the parasite became essentially resistant to atovaquone. The mitochondria from these parasites were still completely susceptible to atovaquone. So these parasites, mitochondrial BC1 complex was completely inhibited, yet the parasites were completely able to survive. So we essentially, by providing a single metabolic bypass, we made them resistant to all electron transport inhibitors working at the BC1 complex, atovaquone, mixothiazole, antimycin, as well as pyridone, acridone, quinolone, and so on. Clearly, of course, chloroquine, artemisinin didn't have any effect on that. So these are the wild type, and this is the DHOD. You can see that they became all resistant to these drugs. So what we showed in this work essentially was that there are 5 different dehydrogenases. All of them actually generate electrons that will convert ubiquinone to ubiquinol, and that ubiquinol needs to be converted back to ubiquinone by the BC1 complex, generating proton gradient and so on. That's the step that is inhibited by atovaquone. Out of all these 5 different enzymes, and 2 of them actually are unique to Plasmodium species. NADH dehydrogenase is a single subunit enzyme, and malate quinone oxidoreductase, which is different than MTH that's present in human. Out of all these, it was only dihyroorotate dehydrogenase that was critical for parasite growth. Okay, that clearly, of course, supports the idea that DHOD is a fantastic drug target, which has been borne out by the work that's been done by Meg Phillips and Prathards and others. So that's the story of DHOD's transgenesis, and that sort of has become a tool for a lot of people. This actually acts as providing this gene in a transgenic fashion makes the parasite resistant to all not only just all electron transport inhibitors, but all the DHOD inhibitors as well. And Suresh Ganeshan, when he was working in the lab, actually showed that this actually is a new selectable marker. We always need selectable markers for doing genetic manipulation of malaria parasites, and this actually added another marker to it. So, as you know, it's the atowacone is not used as a single dose as antimalarial. It is combined with proguanil and malarone. Okay, and so atowacone plus proguanil actually has a synergistic effect on parasite growth inhibition. What we showed, and this was again the work that was done by Indresh when he was in the lab, the proguanil actually does not affect parasite respiration, but at physiological concentration, as achievable concentration, proguanil potentiates collapse of mitochondrial membrane potential, and that leads to parasite device at much lower concentration. So it is proguanil as a prodrug that has this property. Sacroguanil, which is the molecule that is converted from proguanil, which is a dihydrofolate reductase inhibitor, DHFR inhibitor, does not have this property. So it's the proguanil that does that. So we thought that it was the ATP synthase that may be the one that is involved in this alternate way in which to generate membrane potential, and that's why maybe proguanil will be working on that. And that actually got us into working a little bit on ATP synthase complex in Plasmodium falciparum. We had Praveen, when he was in the lab, Praveen Palavaskar and Nina, when he was in the lab, he worked on both on Plasmodium falciparum ATP synthase, but also he did some really nice work on tetrahamina ATP synthase. And it turns out the tetrahamina ATP synthase is really, really quite weird as well. And it turns out that the Plasmodium can give its money's worth as well, I think. And April also worked, April Pershing, when she was in the lab, she worked on some of this aspect as well. So Praveen showed that all the different canonical subunits that were present in Plasmodium genome were all targeted to mitochondria except for C, because of course C is a weird one. It's hard to target that, but all the others were targeted to mitochondria properly. Okay. April actually went on to generate conditional knockdown of beta subunit. So by the way, we could not knock out. So Praveen tried very hard to knock out beta subunit of Plasmodium falciparum ATP synthase, but not successful. So we decided to go the conditional knockdown route, and she generated this GLIMS ribozyme-mediated construct, and that what happened was that by knocking it down to over 80%, over 90% knockdown of expression of the beta subunit of ATP synthase had no effect on parasite growth. Parasite did not care if they had just 10% of the ATP synthase activity that was there, and that was shown by some mutational analysis that April carried out as well. Interestingly, if we had DHOD transgenic parasites, okay, so these are the DHOD transgenic parasites. If you treat them with atowacone, okay, they would continue to grow, okay. But if we added glucosamine at 1.25 millimolar in the same parasites that actually had beta subunit regulated by ribozyme, parasites stopped growing. So essentially, adding glucosamine was just like adding proguanil. So we believe that this gives us a reasonable confidence that it is ATP synthase activity that is likely to be targeted by proguanil. It's something that we really need to show very directly, but it's something that we are working on. So just to summarize this part of the talk is that if you treat parasite with atowacone, the mitochondrial electron transport will be inhibited, and that will lead to essentially no generation of proton gradients coming from these complexes. But if you treat these parasites with atowacone plus proguanil, this will now inhibit, most likely, we think, the ATP synthase And now that will collapse both ways in which to generate electron transport and the generation of mitochondrial membrane potential. And because of that, actually, this could lead to parasite demise. Okay, so I actually have told you a lot about these, but there are also these new drugs that are on the horizon, anti-mitochondrial electron transport chain inhibitors that are on the horizon. In that regard, I've been fortunate to be collaborating with Mike Risco, who has been leading this effort to develop anti-malarial BC1 complex inhibitors. And the one that has really gone further most is this ELQ300, which is a quinolone. It is called ELQ for endocrine-like quinolone. Mike's group has done really a lot of work on this, and this drug actually works at all different stages except for the hypnozoites. It doesn't kill hypnozoites, but it has effect on as a prophylactic drug as well as treatment for malaria as well. It inhibits the parasite electron transport chain at very low nanomolar level, 0.58 nanomolar, compared to human, more than 10,000 nanomolar required for that. So it's really highly selective inhibitor of BC1 complex in malaria parasite. It works against transmission stages as well. Unfortunately for ELQ300, which was actually designated as a candidate by MMB back in 2012, its physical property is rather bad. It's like a brick dust in some ways, and because of that, it was eventually not progressed further. Fortunately, Mike persisted. Mike and his group persisted and developed prodrugs for antimalarials, and the ELQ prodrugs have really enhanced delivery and a single dose cure for malaria. Okay, and the one that is now the most furthest advanced is ELQ331. And it is just as of earlier this month, 331 has been designated as a preclinical candidate, and we hope that it will see first time in human trials fairly within a year or so. So ELQ 300 as a active moiety which can be generated from 331. Essentially, you need to clip this over here, and then you generate ELQ 300. Okay. It's important to know that the prodrug itself actually doesn't have much anti-parasitic activity, BC1 complex activity. Okay, but it clearly inhibits the growth once it goes into the parasite. Okay, so ELQ 300, once it gets converted, it has this effect. Okay, just one more thing about Mike Risco's lab and our collaboration with that lab to show that the ELQ 300 actually works at the QI site. So this atowacone works at the QO site. ELQ 300 works at the QI site, and it's very interesting that while they don't have any cross resistance. So if you have a highly resistant parasite, this is in a rodent system, the ED50 is for atowacone is 0.03 milligram per kilogram, and no recrudescence dose is 1 milligram per kilogram, they become completely resistant by the single point mutation in cytochrome B sequence over here. And there is no cross resistance to that against ELQ 300. And similarly, you have some resistance to resistance that comes about against ELQ 300, but there is no cross resistance to atowacone. So combining a QI site and QO site compounds together will have a really fantastic effect, because parasite will have really hard time generating mutations against both sites. And this would be really interesting to show. And that's something that Alison Stickles and Mike's lab actually showed that by combining them, you actually got no recrudescent parasite coming at all. While combining these 4 doses of atovaquone proguanil together, recrudescent parasites do come up, and some of them actually turn out to have resistance as well, while you don't see that coming up with the combined. So this would be really interesting to pursue further. So the promise of electron transport inhibitors is, I think, significant. Really, it's interesting to look at this further. So one can develop this long half-life which will favor single-dose prophylaxis of treatment, and also we could develop depot injection for long-term protection. Just a single injection can protect you for months together. And these can act as sort of a chemical vaccine. It's something that Marty Smilstein has actually shown to be the case in a mouse model in Mike Risco's group over there. So that's really a great potential for these drugs. Other important thing to keep in mind is something that was shown by Goodman et al. from McFadden's lab, Jeff McFadden's lab, that resistant mutations that actually give you resistance to atovaquone have highly reduced fitness for transmission through mosquitoes. And this will essentially tell you that if you do develop resistant parasites, you are not likely to transmit it to the next people. And this would be an advantage for mitochondrial electron transport chain inhibitors as well. And the inhibition of parasite development actually in mosquitoes can be inhibited by just spraying atovaquone. This was from Flaminia Catoricea's lab. They showed that by spraying some atovaquone on the surfaces on which mosquitoes walk, parasites just do not develop in mosquitoes as well. So this is really exciting potential as well. And as I said, combining QO and QI site inhibitors may be one way to really reduce the rate at which resistance may come about in parasites. So there are clearly questions that are remaining about malaria mitochondria, that need to be addressed. I think there are plenty of questions still remaining. For example, how does mitochondrial DNA replicate and recombine and repair? We have no idea how that happens. It's something that really is worth looking at. Because mitochondrial DNA is really bizarre. In some ways, it is organized in this weird fashion. Its replication is one of the things that happens once resistance comes about. It becomes fixed in the entire population, and the parasite mitochondrial DNA sequence is highly conserved across vast distances, geographic distances. So it's something, how it is done is not really clear. Other thing clearly is that, how do you get the transcription and processing of the different RNAs that are generated from mitochondrial DNA? How do the mitoribosomes or legosomes are actually put together? Something that Hang Jun's lab is actually interested now to pursue. What about tRNAs? There are no tRNAs encoded in the mitochondrial DNA. They need to be imported. If they need to be imported, are the tRNA synthesis imported as well? Well, we don't know that. We don't know how these things are handled. It's something that really worth pursuing as well. What are the essential functions of some of the unannotated plasmodium-specific mitochondrial proteins? We have quite a few proteins that are directed to the mitochondrial compartment, which are essential for the parasite survival, but we don't know their functions. And Ian Lamb in the lab who actually gave a poster earlier today discussing some of these things as well. So and this is important thing to really to pursue as well. And we also would be interested been interested in making row 0 plasmodium falciparum, meaning mitochondrial DNA minus plasmodium falciparum. Variety of different things that we have tried, but we've just not been successful. Why? Why we have not been able to do that? This is in the DHOD transgenic parasites, in which electron transport chain is no longer required, and only 3 proteins that are encoded by mitochondrial DNA are electron transport chain proteins, yet we can't get rid of mitochondrial DNA. So are we missing something about any other function that may be encoded by mitochondrial DNA. It's something that we would like to look at as well. So each one of these questions that I posed over here actually could be, I think, in my opinion, a separate R01. And I hope some of you young people will get on with this. It'll be interesting to see what answers you come up with. So in the last few minutes that I have left, I will just tell you about things that we've been doing with the sodium and cholesterol homeostasis in plasma and phosphorin. There's something that has interested us over the last 10 years or so. And the story begins with this invasion pathway that we all know about, how the parasites invade. They use their own glidosome to drive themselves. It's a really nice talk earlier today on the glidosome and energy generation and so on. Bill Bergman, my colleague Bill Bergman, found out that the myosin heavy chain, the myo-A, interacted with an unusual light chain protein that he called MTIP, or myosin tail interacting protein, that he then, in collaboration with Wim Hall in Seattle, saw the structure. Jürgen Bosch was the lead author of that paper, and he showed that this structure of MTIP required the myosin 15 amino acids at the end, C-terminal end of the myosin, actually went into this pocket over here, this hydrophobic pocket in which it actually resided, that gave a potential to have ability to inhibit parasite growth. So Sandhya Kotakari was the collaborator that Bill Bergman sought out and asked her to design a pharmacophore based on that structure. And she picked up a few compounds, 15 compounds or so, and sent to us, and we tested in our lab for its anti-parasite activity. And one of them turned out to be 150 nanomolar inhibitor. Jeremy Burrows from MMB has told me that this is the most efficient screening that has ever been carried out. After 15 compounds, we got 150 nanomolar hit from that. Fortunately or unfortunately, there were two different types of compounds that Sandhya actually got through iterative screening of her library, her in silico library that she had. And what we found was that they formed into two different groups. So here is the pyrrhazole moiety that can be decorated at a different position by different things over here. And you can see that series A compounds had this kind of decoration, series B compounds had this kind of decorations. And the series B compounds seem to actually be the ones that actually stabilized MTIP and also inhibited sporozoid motility, and potentially they could be the MTIP target. However, these compounds were really not very drug-like. We could not progress them. Erkang Fan at University of Washington and his colleagues synthesized a large number of them, but none of them actually turned out to have really good potential to be developed further. On the other hand, we had these other series of compounds which actually had no effect on MTIP and it didn't look like MTIP was a target for that, but that actually can be made into really very potent compounds and had some really better drug-like properties. So we focused on that a lot more. And that ended up into this publication that came out a few years back on pyrrhazole amide compounds that were potent antimalarials. And it turned out when we got the resistant mutants for these compounds, there are no mutations in the glidosome machinery at all, but the mutations involved, among other things, PFATP4, which was at that time considered to be a calcium pump, but then shown by Kieran Kirk's lab, by Natalie Spillman when she was in Kieran's lab, to be actually a sodium pump. And so these compounds actually work against sodium homeostasis. So the compounds that we got were, this was our initial hit that got 150 nanomolar. No, this is 150 nanomolar compounds. This was the pyrrhazole urea, and this was a 50 nanomolar compound. And from this, these three were then progressed further. Okay. So there are many divergent chemical structures that seem to be working through the same mechanism. They all seem to be targeting this PFATP4, and we'll talk about that in a minute. So here is the pyrazolamide. Here is a spironolone that the Novartis group discovered, and here is DHIQ, dihydroisoquinolone, developed by Kip Guy's group when he was in Nashville. These, see structurally they are very, very different. Not only that, when Kieran's lab actually screened malaria box and pathogen box compounds, 400 plus 125 compounds, about 8% of them actually had the same activity as these compounds, in the sense that they all caused sodium influx into the parasite, meaning they all had the ability to inhibit PFATP4. So if that's the case, if this is the case, that you have such large number of them with about 30,000 antimalarial compounds that were discovered from all these different phenotypic screens, we can predict possibly that there may be a few thousand compounds that may be actually working against PFATP4. Would be really interesting to see what comes out of that. So PFATP4 inhibitors are really very promising because they're the fastest acting antimalarials known thus far. In the clinical trial that was published in 2014, led by Nick White, which showed that these compounds, when given as a single 30 milligram dose orally to the parasite, to patients rather, both Vivax and falciparum malaria patients, they cleared parasites very quickly. They estimated the half-life of the parasite to be less than 1 hour. Less than 1 hour. This is really remarkably quick compounds, much faster than artemisinin as well. So these are really promising compounds, and I certainly hope that one of these will eventually, very soon, will see their widespread deployment eventually. So to look at the physiology of this whole process of sodium, how the sodium homeostasis is maintained, if you look at the parasite growth, as the parasite is growing from ring stage to strophozoites, the amount of the sodium concentration inside the red cell cytoplasm remains about 10 millimolar or so. And as the parasite develops these new permeability pathways and the channels in the plasma membrane of the red cells, sodium concentration starts going up. As it goes up, and the trophozoite stage essentially reaches fairly high concentration up to about 125 to 135 millimolar. And this has to be counteracted by, so all this time, while the red cell cytosolic sodium concentration is going up, the parasite's own sodium concentration has to remain low, and that is achieved by this pump. Okay, so this pump essentially hydrolyzes ATP, and by doing so, it puts the sodium out and exchanging it for the proton. So it maintains a low sodium concentration inside the cytosol, and this particular pump is a type 2D protein. We don't know any structural details of this at this point, but the mechanism for the sodium pumping is likely to be very similar to what you see in other type 2D proteins such as SERCA. Yeah. So the compounds that disrupt sodium homeostasis actually have a really very, very strict SAR. So here are two compounds. One is 92 and here is 122. 92 works as a really, a very high potency as antimalarial growth inhibitor, as well as inducing sodium influx into the parasite, while 122 does not do so. And the only difference between these two compounds is just this. Here is a methyl group, here is a hydrogen. That's the only thing that is different from that, and that actually kills its activity completely. Yet, the sort of interesting part is that yet there are about 30 different chemical scaffolds that are able to disrupt sodium homeostasis. So it's really important that we pursue as many compounds as we can, because they may be acting at different parts of the different positions within the pump itself. So it's something that we are actually actively pursuing and trying to get new antimalarial drugs working on the sodium homeostasis coming up as well. So what are the consequences of high sodium coming into that? And Sudipta Das, when he was a postdoc in the lab, actually carried out some of this work on what happens to the parasite when the sodium concentration goes up into the parasite cytoplasm. So one of the things that found, and this was done in collaboration with Isabel Coppens, who carried out this electron microscopy, that this is an uncontrolled DMSO-treated parasite, trophozoite stage. The same stage parasites in parallel were treated with 10 times EC50 for 2 hours, just 2 hours of treatment. Trophozoite stages start behaving, showing some significant morphological changes, such as constriction of nuclei, formation of segment kind of structures, formation of IMC and ROP trees, and almost merozoite, schizoid-looking parasites. However, they did not progress any further, and most of them actually would continue to die. Okay. So this is something that was seen in just 2 hours of treatment. So here are some other electron micrographs from Isabel that show this very weird morphology in just 2 hours of treatment with this compound. This clearly is telling us that some signaling events are occurring in the parasites, and we consider this to be premature signaling of schizogony, and what that might involve, what kind of signaling involved is something that we are interested in. Aarti Ramanathan actually gave a poster earlier today talking about how phosphoproteome seems to change dramatically in just 2 hours of treatment with these drugs. Okay. So other thing that we found was that if we made after treatment of the parasite with these drugs, if we prepared the saponinized parasite pellet, the amount of protein that was present was highly reduced. And that's something that Joanne Morrissey came into my office and showed me that, do you see that this pellet is smaller? It sure was smaller. It turns out that if you do the same kind of releasing of the hemoglobin from the red cells, instead of saponin, if you use anthracycline O, you don't see the release of the parasite protein. So there seems to be a saponin sensitivity that is induced by these parasites, but it does not inhibit protein synthesis as such. So what we went on to show that this saponin sensitivity is dose dependent, and it happens with, the way we look at it by saponin treated parasite, whether or not they would have aldolase associated with that, we can see the very low concentration of the compounds. Just 2 hours of treatment causes saponin release from the parasite. And this is due to cholesterol. We showed that by carrying out MBCD treatment and so on, to show that actually it is incorporation of cholesterol. So those of you who may not know this, but the saponin is a cholesterol requiring detergent. It forms pores only when cholesterol is present in the plasma membrane of the cells. And these pores then allow the cytoplasmic proteins to leak out. So other thing we found out that this was actually reversible. So here is treatment of the parasite, the cytosolic leakage that you can see. And if you wash the drug off, it actually comes back. So the aldolase remains intact. So the whole process seems to be reversible, and there must be an active process of cholesterol exclusion that may be occurring in the parasite. So this is something that's interesting. So this is what the parasite looks like. The parasite in general has a lot of cholesterol in the plasma membrane of the infected red cell. There is significant amount of cholesterol that is present in the PVM, but there is very little, almost no cholesterol present in the plasma membrane of the parasite, and that allows you to do the saponin lysis and getting all that stuff that you do with the biochemistry of malaria parasites. If you treat them with the drug, with the PFATP4 inhibitors, that now leads to cholesterol coming into the plasma membrane of the parasite, and that leads to leakage of the parasite cytosol upon saponin treatment. So this actually led us to yet another branch of things to do, and that was in collaboration with Dan Goldberg's lab, with Eva Esteban, and Sudipta Das when he was still in the lab at that time. And what we showed is that there is yet another protein that seems to be important for that. So there are these three compounds that Eva and colleagues had found that they seem to the resistance to these compounds seem to affect this particular protein. Is called we are now calling this NCR1, Niemann-Pick type C1 related protein. The mutations actually occurred in that protein to give resistance to these compounds. And this was initially annotated as a sterile proton antiporter in Plasmodium. And when we saw this, when Eva actually presented this work at MPM a few years back, we immediately struck up a collaboration with Dan and Eva, and to find out what this particular thing might be, whether or not it also induced the cholesterol homeostasis gene. And sure enough, inhibition of this particular pump by 9108 or this compound, three different compounds, they all lead to saponin sensitivity of the parasite. And not only that, that saponin sensitivity was actually reversible. So if you wash the drug off, the saponin sensitivity was eliminated, and the parasite became resistant to that. So all this stuff actually led to oh, by the way, other thing was showed that it was not sodium dependent. So these compounds had no effect on ATP4 as a target, as a sodium homeostasis at all. It was independent of that. So this sort of interesting aspect of that. So this allowed us to really look at malaria box and the pathogen box compounds distributed by MMB to see what compounds may actually have the same activity as the NCR1 inhibitors. And this work was done by Suyash Bhatnagar and Cezanne Nicholas. What they showed was that they developed a sort of a high throughput assay for that, and they showed that there were compounds that were clearly sodium influx dependent. Okay, so all the green ones are the ones that were shown by Kieran Kirk's lab as the sodium influx and saponin sensitivity due to sodium influx. But there were compounds that were not identified in Kieran's lab as inducing sodium influx, but still cause saponin sensitivity. And these, we believe, are the inhibitors of NCR1. And this was shown several different chemical scaffolds, really many different chemical scaffolds that actually showed this to be the case. This is all published, so you can look it up, these structures. So again, many chemical scaffolds, they seem to be affecting both ATP4 as well as NCR1. I think we may have some things to mine in the collection of compounds that we have. So just to very quickly tell you about cholesterol and malaria parasites, know that I've gone over my 1 hour time slot here, but just to tell you a little bit about what we know. Accessible cholesterol, so there are two pools, there is accessible and inaccessible cholesterol. We believe that accessible cholesterol in the RBC membrane is essential for parasite growth and development. Removal of this accessible cholesterol from red cell membrane results in dramatic expulsion of the trophozoites from the RBC, something that actually was shown in Kasturi Halda's lab some years back, and we have actually carried out followed this up in some detail now. The treatment with these inhibitors, so PFATP4 and NCR1 inhibitors actually inhibit this expulsion, so pointing to this involvement of this active transport of accessible cholesterol in Plasmodium falciparum infection. And we know that actin depolymerization does not inhibit cholesterol depletion mediated extrusion of parasites. All this stuff will be described very nicely in a talk that Avantika here from the lab will be giving tomorrow. Please attend at number 76. It will be something that will be interesting to look at. So I come to the end of my talk. I have to thank a lot of people, and I will not take a lot of time to do that. But this just amazing journey of 50 years or so that I've been blessed with. we have collaborators at Drexel, outstanding colleagues. Joanne is the pillar of my lab. She has been working with me since mid-80s. It's over 30 years, Joanne. She has been just fantastic. And Mike as well, who's been with me for a very long time. We've been colleagues for a long time. All the people that some of them I've mentioned, some of them I have not mentioned, please forgive me. I don't want to take too much time here, but I should not let Tom Daly, Jim Burns, Bill Bergman, Sandhya Cordigary. Tom is retired, but Jim, Bill, and Sandhya are still there. Well, Bill is retired. Bill is retired as well. I should put an asterisk there, just retired. But these are outstanding colleagues that have been really, in spite of having changed, changed names of place and all the different things that went on politically in our university. It's these colleagues that have kept me engaged and energized, and it's been really fun. Many collaborations all over the world, and I'm really thankful to all of them. Finally, of course, funding. I'm thankful for the generosity of U.S. taxpayers, for supporting all the work that we've done for all these years, and all the study section members who thought that what we were doing was important enough, so that they give us fundable scores. Not always, but many times they were sufficient. So, as well as funding from NSF, WHO, Burroughs Wellcome Fund, and MMB as well. So, thank you for doing all this, the funding for us. I have had really, so this is my lab. We just went on an axe throwing competition. This is the current lab right now. We have lots of fun. And the list of all the students and postdoc fellows who have been working with me all these years. Prema Arasu was my first graduate student, 1985. She retired just recently as a vice provost from Kansas State University. I'm still continuing, but she is retired. And now here's the latest student, Swaksha Rachuri, just joined my lab. I don't know why, but she has just joined my lab. Postdoctoral fellows, and many of them actually are former graduate students. Some of them stay on for a year or two, and others continue for a longer time. So some of them are listed on both boxes over here. And finally, all these things would not be possible without my family. This is Sheila and my boys, Ashish and Avinash. This is, you can imagine this a few years back. Boys are now in their 30s. Sheila held the fort. Without that, I would not be able to do all the things, in spite of her own faculty position and all she had to do. And the boys, gave me permission to do things what I needed to do. And finally, the Vaidya clan and Nayak family. So thank you very much. And I would be very happy to answer questions if there are any. Thank you, guys.
Well, thank you very much, Akhil. Just wanted to check because I changed microphones. I assume you can hear me.
Yes, I can.
I think others can as well. There are a number of questions that have come in, and one of the disappointments of a Zoom meeting is the constraints that it puts on discussion time, both of course in sessions because we need to keep to time and through lack of virtual interaction. So. I think, assuming you're willing, it would be good to be able to get through some of these questions to extend our discussion time until 6 o'clock or so. Let me remind all of those who are observing this talk that while we have several hundred participants in the talk, too many to turn on cameras and have people speak individually, I will moderate the discussion and read out the questions for Dr. Vaidya. You are welcome to type your questions into the Q&A. For those who are watching on YouTube Live, if you would like to email me questions to my email address, my address is droos@upenn.edu, 2 N's on the upenn.edu, and I'll be happy to convey any questions that come in over YouTube as well. So let's start at the top. I do note that Luisa Figueiredo from Portugal notes that she's happy to be able to see some toxoplasma parasites in the background. To be honest, that's not exactly what she said, but I thought I would change my virtual background to a different picture of toxoplasma, since she's not actually online to be able to ask. But she did have a question as well, asking whether it would be interesting to study how the interaction of the mitochondrion and the apicoplast in Plasmodium, especially noting that the mitochondrial and nuclear DNA are synchronized in trypanosomes. Is there synchronization between the nuclear DNA, mitochondrial DNA, and apicoplast biology in Plasmodium?
Luisa, thanks. I think we have always wondered that there's something going on between mitochondria and apicoplast. They are very closely attached together. But then, of course, Ellen Ye and Derisi came around and knocked out apicoplast completely, but they're perfectly fine, and their mitochondrial physiology is perfectly fine. So if there is any connection between mitochondria and apicoplast and their physiology, it's something that we don't know about. We don't know what it is. On the other hand, even in the so-called apicoplast minus lines, there are these remnants of apicoplast still remain, apicoplast-looking organelles still remain. Whether or not they still provide something to the mitochondria, we don't know. It's really an important question, but we don't have any good answer to that at this point.
And is there any association between the mitochondria and the nuclear replication, as there is dramatically in kinetoplastida?
Right. So in kinetoplast, clearly the kDNA replicates first, and then the nuclear DNA, and so on. Or here, they seem to go about the same time, but I think mitochondrial DNA starts replicating earlier. So you can see the mitochondrial DNA getting a larger amount in trophozoite stage, where you still don't have all the replication. So schizogeny is very different from just the nuclear division that you see in kinetoplast.
Yeah, I mean, it's interesting in toxoplasma, mitochondrial replication proceeds well after the separation of the nuclei, but we don't know about mitochondrial DNA replication. I don't believe that that's been looked at.
Right.
Morphology is, of course, quite different. Meg Phillips has an interesting observation triggered by your comment that a colleague had told you to work on something important. That's exactly. Excuse me, just a moment. Triggered by your comment from your colleague that you should work on something important, she remembers that that's exactly what you told her when she was a young assistant professor. For those who don't know Dr. Phillips, I should point out that Meg is currently the chair of the Department of Biochemistry at University of Texas Southwestern Medical Center in Dallas. It's that work that inspired her to start a malaria project, working on DHODH and leading to DSM 265. This was just a comment from Meg, but I wonder, especially in light of the large number of junior scientists participating in the meeting, Akhil, if you'd maybe care to comment on that. You have worked on the projects you've worked on, driven, I think, by intellectual interest, but always with the hope that that work would lead to something that is practical and important. And so the question is, do you have any insights or guidance or comments as to how what seems like peculiarly abstract molecular biology of a tandemly repeated DNA in Plasmodium parasites could have led us to drugs that are now on the verge of clinical trials? Are there comments that you would add about how to pick projects from your list of interesting, outstanding questions?
It's really important. I think that three things go together. First is, of course, it's really hard to work in science. I mean, it's hard work, right? So you need to, really go for the jugular. Don't fool around with capillaries. That's one thing. Just go for something that you think is very important. And second is, when in doubt, this is something that I always feel really is important. When you have some doubts in your mind, go and pray to Darwin. Darwin always gives you the answer. Evolution is really important. It's something, if something that is highly conserved. So the reason why I thought that those the 6 kb mitochondrial, the 6 kb DNA was important is that it's highly conserved. That this has to have something important, and I couldn't figure out because at that time, you couldn't do sequencing that easily. It turned out that with mitochondrial DNA, but so yeah, if something is conserved or something is present only in certain branches and not in others, those would be the kind of questions that I would ask first to see whether how important an area of work is. Yeah.
Thank you. Setu Nair asks why atovaquone-resistant parasites are not transmission competent.
A very good question, Setu. The reason that I think, and this is something that we showed in 2005 in Mike Mather's paper in JBC, when we engineered Rhodobacter capsulatus in collaboration with Fevzi Daldal at University of Pennsylvania to resemble Plasmodium cytochrome B sequence. And when we did that, it actually became more resistant, more susceptible to atovaquone. And then when we made these mutations that give you resistance to atovaquone, they became resistant. But not only that, the wild type bacteria, Rhodobacter capsulatus, had a fitness defect. And that fitness defect, Fevzi Daldal's lab finally went on to show that is because of these mutations lead to formation of or stabilization of or not stabilization, release of semiquinone, single electron reduced ubiquinone called semiquinone to come out of that and cause oxidative stress. And that becomes more important. There's more oxidative environment. And in the insect stages, this may be more important for that. And that may be the reason I think there is a fitness cost associated with resistance development. That's really important. Yeah, good question.
Interesting. There are a number of questions about drug targets and resistance mechanisms. I'll come back to those for a moment, but I'd like to take a question coming in from the YouTube observers. There's a question from Smita Bhaduri McIntosh asking two questions, actually. First of all, whether you have any idea of the composition of the replicosome that's responsible for 6 kb DNA replication in the mitochondrion.
Sumita, come back to the lab. No, we don't. And it's something that we very much would like to do that. Yeah, no, Sumita was a graduate student years back. It's an important point. As I said, that was one of the things that we have as an open question. Yeah.
Yeah. Smita also was interested in whether you had any thoughts on the potential for ETC mutants as live attenuated vaccines, whether those might act as with transmission blocking potential.
Well, I mean, I think I would be, it's the sporozoite stage that seems to be the important part of that. We can get attenuated sporozoite vaccine. There are much better attenuation protocols that the people have developed for sporozoite than I think the ETC. I would not choose ETC mutation as a first choice for that now.
Elizabeth Winsler asked if there's genetic evidence that proguanil works via ATP synthase.
It's only indirect evidence, Elizabeth, that we showed, by having the knockdown thing. We don't have direct evidence. We don't have a resistant parasite. A group in Australia has proguanil resistant parasites, and they said that they actually did not see any mutation in ATP synthase, but I have not seen all the data from that, but that's what I've heard through the grapevine. But the question is still open as far as exactly how proguanil works. But in the case of mammalian system, there has been some evidence for it working through ATP synthase, the proguanil working in that as well. Yeah, biguanides, overall biguanides.
And Pradeep Rathod was asking whether we know whether the conversion of the ELQ331 prodrug into ELQ300 is encoded by the parasite or the host, which could, of course, have implications for resistance.
Right. So the conversion seems to be, as far as I know, is through an esterase, and the esterases are present in large amount in the bloodstream. So from what I've heard from Mike Risco's group is that when you give 331, you almost you don't detect 331, the prodrug, almost negligibly. It's not present at all. So it gets converted very quickly to ALQ300. It's done by the host enzyme. The parasite enzyme probably is not important. In culture, it's probably the parasite enzyme, or there may be esterase that may be present in the red cell that actually activates 331 to 300.
Mm-hmm. Manuel Linas asks whether it might be possible to generate petite mutants, mitochondrial minus mutant parasites, if you provided acetate as a complement.
Manuel, that's a good question. We have talked about acetate complementation in some ways. We haven't done that. We haven't done that experiment. We can try that. I have a feeling that there's something else going on, but you may be right. Maybe acetate might help. That's a good suggestion.
Experiment to try. It'd be pretty exciting. Yeah. There's certainly the discovery of petite mutants in yeast has been pretty revealing. Yeah. And Michelle Klingbeil is asking whether there are non-coding RNAs generated in the mitochondria that are essential.
There are so many small RNAs, Michelle, that are encoded in encoded by mitochondrial DNA. We don't know the function of all of them. If you look at that, there are multiple copies of them, and they're present in large amount. But most of them actually supposed to be ribosomal RNA pieces. But there are other things that may be in there as well. Now, we know that in human mitochondria, in vertebrate mitochondria, mitochondrial DNA, ribosomal RNA actually has open reading frames, short open reading frames that are translated into peptides. And these peptides have biological, physiological activity. Some of them actually, there's something called MOTC, which regulates insulin sensitivity and has what people call mitokines. So these are encoded in the mitochondrial genome on ribosomal RNA, short pieces like 16 amino acids or so that can do this thing. So that's something that I've been playing around with a little bit. There are about 18, so there are about 92 open reading frames. If you don't count AUG as the start codon, you can look for how short peptides you can generate if you have a non-canonical initiation. There can be 89 different peptides that can come out of that. It's a little large number to really see which one is real or not. But I looked for it in the proteome data, and we don't see any of those things in the proteome at all. But you don't see cytochrome B either in many of these things as well. So I'm not completely convinced that they are not there. But it's something that's worth looking at. Yeah.
Svaksha Rachuri asked, who I should say is a first-time MPM attendee, wanted to ask about cholesterol pathways, in particular, whether the removal of these compounds to reverse the process, what happens to the cholesterol?
So when you wash the drugs off after treatment, the cholesterol goes into the plasma. It gets out of the plasma membrane. It's no longer there. It goes back to wherever it's supposed to go. It goes back to, most likely going back to PVM and then back to PPM, red cell plasma membrane. So it's something that you may, Svaksha, she is a MD PhD student in the lab. She should attend Avantika's talk tomorrow. So Avantika will talk about some of that.
Ian Lamb asks why it is that with all of the high throughput screens that have been done against Plasmodium to identify novel antimalarials, we've seen such a paucity of targets, especially given the diversity of the chemical compounds and the large number of genes that we know are essential for parasite viability.
Great question, Ian. I think it's something that Elizabeth Winsler and Meg and others can tell you as well. It's true. There are all these high throughput screens that Elizabeth has been involved in that clearly, and they have a limited number of targets that they come up with. Perhaps it's the way in which we do the screens. Perhaps we're doing growth inhibition curves as a way to really screen for that. But it is a conundrum. It's not very clear why we haven't identified many more targets than what we have. But clearly, there are certain targets that are highly druggable, and those are the ones that come up. BC1 complex, DHOD, the ATP4, and so on. Those come up again and again and again.
And Innocent Safuki asks whether you think the pyrazolamide effect targets mechanical properties of infected red cells and contributes to retention in the spleen. This could potentially provide an explanation for the very rapid clearance that you observe.
No, it's absolutely, that's what we think is going on. Something is changing in the infected RBC, which then it could be, there's some reports, published reports saying that the rigidity is increased after treatment with 609, which is the spiroindolent compound. There are also potential evidence for other modifications that may occur as well. Something called eryptosis, which is Kip Guy's lab has suggested as well. So changes in the red cell, what exactly those changes are is not really clear, and something we have some ideas about the potential role of cholesterol in there as well. But yeah, clearly that may be the reason, because in vitro, in culture, these compounds don't kill that fast. They don't kill fast. It's about 35 hours or so to get 99.9% clearance. But in vivo, it's half-life is 1 hour. It's just remarkable. Yeah.
Tanashekaran Shanmugam asks whether heme scavenged from hemoglobin, whether heme is scavenged from hemoglobin in the heme pathway mutants.
That's the idea. That's what you have a very small amount of heme that is needed in the blood stages, and there is enough of that actually maybe hanging around. And Joaquin Nal's group actually has shown that the amount of free heme is sufficient for it to be salvaged. How the salvage occurs is very important, because the salvage will require enormous amount of care, because heme is a very reactive compound. It needs to be properly chaperoned and so on. We know that there are transporters and all that for those organisms. For example, none of the nematodes actually have heme synthesis pathway. They have to salvage it, and that salvage occurs through very elaborate process that Iqbal Hamza and others have actually shown how that works. Whether or not Plasmodium has those kind of salvaging things or not is not clear. It's another open question, not clear. Danny clearly was involved in the heme thing many years back. He was the one who actually, one of the people suggested that heme biosynthesis may be a great target. Sorry, Danny, it doesn't seem to be.
So we're coming up to the top of the hour when this meeting is scheduled to end. I think it's only appropriate to give the last question to Joanne Morrissey. But before I do, I'd like to thank Akhil very much and comment that for all of those who've entered questions or comments on the Q&A or via email, I will pass those along to Akhil later on. And also mention that as friends and colleagues of Dr. Vaidya, or many of them know, we're hosting a virtual reception via Zoom following the close of this seminar. I apologize if you did not receive an invitation for that, but are a longtime friend and colleague and would like to join. If that is the case, feel free to send me an email again to droos@upenn.edu, and I will make sure you get that invitation. Actually, maybe, I'm sorry, to really be fully gender balanced and whatever, I should give final questions to both Mike McIntosh and Joanne Morrissey. Mike asks, what's known about the structure of mitochondrial ribosomes, polysomes, and ribosomal import?
What's known about polysomal import, Mike?
No, but he's asking about what's known about the structure of the mitochondrial ribosomes and polysomes, and also about the import of ribosomal proteins that are, of course, necessary for translation.
It's clearly nothing. Not much is known at all. So I think it's something that we clearly is, and one more thing that we need to add on to the questions remaining.
And just to cut you off, because we have just 1 minute remaining, Joanne asks to ask you about your dinosaur egg experience.
No, so our lab, this our standing joke is that I always tell that a dinosaur egg, if you find one, you have to put it in the incubator to see if anything hatches at all. And most of my ideas turn out to be like those dinosaur eggs, fossilized, don't mature into anything. Occasionally something matures out, and that we follow through. Thanks, Joanne.
Well, thanks very much. Thanks to all of the participants, even at this late hour here and an even later hour for those who are calling in from Europe or India or East Asia. I'll note that there are more than 200 participants still remaining on this seminar, a Lilly auditorium full of people who unfortunately can only join you virtually. But let me just ask for a round of virtual applause from everyone. I will look forward to meeting with friends and colleagues of Akhil on the Zoom call after I go to grab myself a beer.
Okay, and are you going to send me the chat? Are you going to send me the chats?
I'm sorry, yes, I will send all of the chats and
the question answer.
I've made copies of all of those and we'll send those all to you later on this evening.
Good, thank you.
Thank you once again,
thank you
Akhil,
all.
and goodbye to everyone.
Thank you.